All right, so what we're going to do is go through the activity that you guys will be doing while I am on my field trip with my anatomy classes. So, what we're going to do in biology today is work through this sheet here. So, it's pages 27 through 29. Okay, so you will end right here. What I'm going to do is just kind of slowly go through this so that you guys will then have the rest of the time to get it done. So it says, below are two partial sequences of DNA bases shown for only one strand of DNA. Sequence 1 is from a human, and sequence 2 is from a cow. In both humans and cows, this sequence is part of a set of instructions for controlling a bodily function. In this case, the sequence is containing the gene to make the protein insulin. Insulin is necessary for the uptake of sugar from the blood. Without insulin, a person cannot use digested sugars the same way others can, and they have a disease called diabetes. So on page 1, these are your instructions. What you're going to do is take this DNA strand and first break it up into the triplets, right? So here's a triplet, here's a triplet, right? All of these are triplets. So what I want you to do is draw a vertical line in between these. Okay, and this is on my key, so if you'd like a reference, the sub should have that for you. What you will then do after you have broken these into thirds is write down what the mRNA strand would look like. Okay, so this is DNA, and we're saying we're going to go through transcription, and we're going to take the DNA and turn it into the message, the messenger RNA. So remember, you're only using A, T, but we don't use T. We change the T to a U. So we have AUCG in mRNA. DNA is ATCG. So you've broken it into thirds. I would then like you to write down what that transcribes into. So CCA, well, that would transcribe into GGU. Okay, so you would write that down. And then you can write down the second one, TAG. What does that turn into? Well, it'd be AUC. So you'd write that down. You should have a total of 9 codons made, so 27 total letters. After you have done that, you will then take that code you made and turn it into amino acids. So that's when you'll use the wheel chart. You'll take what you made here and then use the wheel chart and write down the amino acids made. So you should have a total of 9 amino acids. You will then do the same exact thing with the cow, right? Take its DNA, break it into thirds, and then transcribe it, and then translate it into amino acids. The top of 28 is then asking about the amino acids you made. So it says comparing the gene to the human gene to the cow gene, how many of the codons are exactly the same? So for number 1, you're going to look at the mRNA strands and figure out how many codons are similar. You will then look at, for number 2, you'll look at the amino acids and say, okay, how many amino acids do I have that are the same? Okay, so you should see that this number here is slightly smaller than this number here. Okay, that's your hint. Next, for number 3, you're going to look at your wheel chart, and wherever you see leucine, I want you to write in the code. You should have 6 total codes written in here. This question here is a critical thinking question. Okay, so what it's essentially asking, if I were to simplify it, is can you have 2 codons that are different but make the same amino acid? Okay, so that's for you to write down. Here, we are then going to look at a mutated gene versus a normal gene and see whether or not they make the same exact amino acid. So it says diabetes is a disease characterized by the inability to break down sugars. Often a person with diabetes has a defective DNA sequence that isn't able to code for making of insulin protein like it should be able to. says, suppose a person has mutation in their DNA and the first triplet, so their first codon for insulin reads TAG, or TAT, my apologies, TAT. The normal version is TAG. And it says, what amino acid does the mutant DNA and the normal DNA code for? So you will take the TAT and first turn it into the RNA version. So TAT, if we were to go through transcription, would be AUA. Then you need to look at what amino acid does AUA make. So look at your chart, and you should have isoleucine. Okay, I'll give you that one. You then are going to take this code, TAG, and do the same thing, where you turn it into mRNA, and then you're going to write down the amino acid. Then it says, will the person be diabetic? So what that is asking is, if the mutant amino acid made and the normal amino acid made are the exact same, okay, they both make the same exact amino acid, then we wouldn't expect any mutations, right? Because if you make the same thing, then you should be fine and make the same protein. However, if the mutant and the normal are different, then we would expect that this person is diabetic because they're not producing the right proteins. Number 6 wants you to then look at a new code, TCT. Okay, so we know, now you will know what amino acid is made from the normal one. So it wants you to compare again. Okay, TAG is still the normal, and you are going to compare the amino acid made by TAG versus TCT. But remember, first you take these and turn them into RNA. So TCT would be AGA. So you'll say, what does AGA make? And then you need to look back and say, what did I put down right here for what TAG made? And once again, if they are the same, then we would say the person is not diabetic. If the two amino acids made are different, then we would say they're diabetic. Okay, last thing here. Now what we're doing is we're given a human DNA strand, chimpanzee, pig, and cricket. And we're going to do the same process we did up here on 27. We're just going to do it for 2 more organisms. However, the code's shorter. So that's how we make it easier on you. So we take CCA, TAG, CAC, CTA right here for the human, and we'll write down what it transcribes into, and then we'll write the amino acid sequence made from that. You'll then do that for the chimp, the pig, and the cricket. Once you have that, it's going to say, look at what amino acids are made from these 4 organisms and tell me which 2 are most closely related. The 2 that are most closely related would make the most similar amino acid sequence. Okay. Then it wants you to do one last thing. An unknown organism is found in the forest, and the gene is sequenced as follows. So you need to find out what this unknown gene transcribes and translates into, and then based on the sequence you made, I want you to tell me what animal does it closely relate to of the 4 options. Once you've done all that, then you are done for the day. As always, if you have questions, just email me, and I will write some notes in for the sub as well, and she will have the key, or he or she, whoever it may be. Okay? All right.